Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry, brought to you by Beckman Coulter at Bite Size Bio.
Hi, welcome to Flow Stars, a very special edition recorded live with a live audience at CYTO 2023. We feature Jessica Houston, Paul Wallace and Pratip Chattopadhyay, and we talk about where they see the utilization of flow cytometry going over the next 10 years.
It's a hard question, but I think that it's probably going to stay the same because we're going to have more technologies to choose from. And so there's going to be a little bit of a drag on the number of people doing flow. But the flip side of it is that actually doing a flow cytometry experiment is infinitely easier than it used to be. And data analysis is something that, we're all focusing on. And so, I think that's going to make it easier to take up flow. So those two trends are probably going to cancel each other out.
Including different opinions.
I don't think there's any question that the utilization will increase. We do see changes. We see, maybe CD4s are no longer needed as much as they were. There's definitely a transition with MRD going more to next-gen sequencing. But there are always new techniques coming out, new applications coming out. And, we're moving into a world of microvesicles. And I just see that as continuing on for a long time.
And is conventional flow cytometry dead?
No, it's not dead. We had a good example of at the meeting with resource poor places where it's so critical to still use it. So, the complexity, spectral, whether you want to say it's complex or not. There's still a lot of places in the world that need just, the standard, simple method.
All in this very special live episode of Flow Stars.
Hey, ladies and gentlemen, we're about to start. First off, I'm going to say a quick thank you to Beckman Coulter for not entertaining you this evening and letting flow cytometrists entertain you instead, which is a bit different, isn't it? Okay, so it is a live Flow Stars, and we've got three very special guests tonight. We have Paul Wallace, Jessica Houston.
All right, Jessica.
And Pratip Chattopadhyay, even. Jesus, fashion name, mate. Okay, so I'm Peter O'Toole from the University of York. I am not working for Beckman Coulter. Okay, so don't get that confused, please. Okay, so let's kick off. I'm going to ask our guests different questions. You have also messaged in questions. Okay, so some of those questions, I will call your name out, and I will say, actually, if you're embarrassed by your question, tough luck. Okay, so my first question is, which company does the best CYTO party?
Oh, boy.
I'm going to start with Paul.
Peter, I have to say I love them all. There are definitely a lot of good parties, but there are a lot of good parties at CYTO, but the company parties are the best.
Beckman Coulter.
Now, Jessica, I'd just like to remind you this is being recorded.
Yes, okay, I know.
Would you like to say
Be
anything
more
else?
diplomatic about that.
Jessica is the next president, so really she should say that ISAC throws the best parties.
Yes.
And
I don't know about that.
I don't have any conflict of interest, and you've put me up here. I can say that Coulter throws the best parties.
There you go.
So.
Okay, so the next question actually comes from Johnny Moore.
Uh-oh.
So Johnny asked, what is the best CYTO that you've actually attended?
Oh, the best.
Oh, I'm going to start that in this time. Pratip.
So I have, I have a selfish answer, but Vancouver was my favorite.
All right.
We managed to pull together a meeting that was heavily focused on single cell biology. And, that's who we are. And it was so great to see it in different ways. And Vancouver is really pretty. So this is probably my second favorite.
Wow, that's a good one. And I have to pull the selfish card as well. Montreal. This one is my favorite so far. I feel like, as
What was that, Jessica?
This one.
This one. Yeah, this one. CYTO. Obviously it's not over yet. We have one more day to go, but
we have a party to go.
And we have a party. Yes, we do. But
Should we go on?
I think this one's gone well, and I'm really happy about it, and I'm glad that all the content we have in the plenaries and have been well received. So yeah, I have to go.
So, Jessica, who's in charge of this one?
That would be me. Yeah.
There's no conflict here, is there at all? So, Paul, now you know that Jessica's in charge of this one.
What I know is that Johnny asked the question, so it's Philadelphia, which is my hometown. But I think outside of Philadelphia, which would have to be number one, it would be Quebec City. That was a fabulous one, and Montpelier. I mean, I like the Montpelier ones, but Seattle, which is the one that I hosted, would be one.
Okay, so actually another question that came in is, why do you keep coming back to CYTO? I'm going Jessica first.
ISAC is my home. It's my scientific community, and the CYTO meeting is so pinnacle in for the society. And I obviously have an obligation now. That's why I'm coming back. But it's
You
it's
feel
my home.
obligated, huh?
Yeah. All of the work I've done through the society and the meetings and the contributions we made in our lab to the meeting is, it's a hard question. I just come back because this is a home. It's a community. It's family. Yeah.
Okay, and Paul?
Well, I mean, I personally love the plenaries and all the sessions, but I think the reason I come back is because of the people. I come back to see you guys. I mean, it's a lot of fun just reconnecting with a lot of old friends.
Everyone, or is there someone who you'd rather not see?
Everybody here.
Pratip, now you're now here. You're now going commercial.
Yeah.
So does that make it different coming to CYTO?
So it totally makes it different. My experience has evolved because first it was really to scratch the itch to teach flow cytometry, and I got to do that in a forum where people really appreciate education and teaching. And so, that was really exciting and, kind of empowering to me. And then it was, to present my cytometry work. And so, and now it's become more to come and connect with vendors and people doing exciting new science who are in the commercial space. And, I'm almost embarrassed to say that I'm spending more of my time, planning out new collaborations and new partnerships with commercial entities rather than going to sessions and plenaries. And so I got to figure out a way to clone myself, but maybe we'll have a new company around to do that. So,
Pratip, thank you. So a question from Chris Hall. Now this is got to be the best question that I actually read this morning. So I recently discovered, said Chris, that the acceptor in tandem dye Pacific Blue is a sunscreen. Have you ever tried adding sunscreen to cells to analyze on your instruments? And that's a great question, wasn't it? And what other household items have you run through your flow cytometers?
I have a story about this. So my neighbor there was a leak coming from his yard that was totally flooding our yard, and I was convinced it was from his pool water. And we had just gotten our CYTO a few years ago, and so I quietly took a sample of the water and I ran it on CYTO, with a control, rainwater control and a tap water control, and compared it, and I couldn't tell what was going on. So pool water that I thought, that allegedly my neighbor dumped into our yard.
Paul, Jessica, household items you put through a flow cytometer?
I don't
Well, we
think
all
I've done
put
that.
we all put bleach through it, but I guess the oddball ones was milk. I was trying to use that as an absorbent to get rid of some autofluorescence, and Coca-Cola.
Oh.
Coca-Cola.
I was just curious as to what it would do. Didn't do much.
How many times did you have to prime or purge to get the air out after?
No, it's actually it's a cleanse. I mean, all those bubbles.
Interesting.
Oh, that's just bizarre. Okay, next question is from Claire Brown. Okay, What was the most challenging career transition for you? So I might start actually this time with Pratip.
It was they were all challenging. Leaving Mario's lab was, like growing up and leaving home, so it was that was definitely hard. But I think the hardest transition was, running my own business, because then, it was a small business. I was cleaning toilets. I was, doing everything. And it's nice not having a biosafety committee or, purchasing group or something like that. You can do whatever the heck you want, but it's also taxing, and it gets exhausting really quickly. So
It's cleaning toilets. It's like, oh, yeah, I've even got to clean the toilets. Now, if you ever came to University of York, I wish I was allowed to clean the toilets.
yeah, really.
Because I do a lot better job than the cleaner does. Trust me, it just agrees me. I can't do it. I could do it if I was allowed to. It's not like I can't clean the toilet.
You can come to my lab and clean the toilets. So
Gosh, have we got a job for you?
I'll get the marigolds. Jessica, difficult transition time.
For me, it was definitely postdoc to faculty position. That's, you become a faculty, and it's a bit of a deer in the headlights experience. But the reason why it was most challenging is because when you're postdoc and you decide to start a research program, you wonder, what am I going to do? And it was, should I take what I'm doing as a postdoc and continue? And I came from Los Alamos National Lab. We were developing that technology in flow and brought it to NMSU. And I talked to the guys there, and should I do this? It's like, yeah, why not? And for me, it was like, I didn't translate that, yeah, this is my work. I can continue it and maybe bring in new aspects to it. But that was the hardest part until I got a comforting, do it. Mm-hmm. Yeah.
I think most people can empathize with that. And Paul.
So as you all know, I'm a little bit senile, so I only can think about what is the most current.
Would you like me to repeat the question?
Yes, that might be a good idea. But as I went into retirement, so announcing that I was going to retirement and then no longer being relevant to the organization or to the staff was very difficult. But actually retiring and then having not a whole lot to do, and fortunately, I flunked retirement and then joined Cygro. So I think the flunking of retirement was probably the hardest transition.
Okay, so that brings me really nicely onto one of the questions I had for you as well. Was when you do properly retire, will you ever look at a flow cytometer again? I'll start with you, Paul.
I've always wanted to have one in my basement.
To cleaning out your Coca-Cola?
There's just to run things through. I mean, I couldn't imagine pollen, microparticles. I would love to have one in my basement. So, Beckman, Coulter, if you've got any things that you want to use as anchors, I got a place to anchor it.
Jessica.
No, instruments for Paul.
Yeah, one of these days when I retire, I would like to have some lasers hanging around, and maybe play around with that. Maybe not a flow cytometer, but things I can tinker with. That'd be fun. Yeah.
So just lasers? Yeah. Are you going to put
lasers,
some smoke in the room and then just someone sits around?
Just to shoot at the planes? The planes that fly overhead?
I don't know. Something like that. I don't know.
So, I remember when I was in graduate school, there was a professor named Arthur Dannenberg, who was in his 90s, toddling around the hall doing TB work. And so I told my wife that I'm going to Art Dannenberg it. I'm just going to go right to the end. My bony body is going to have, a lab coat barely hanging off of it, and I'm going to have a blast, People might think I'm crazy, but, who cares, right? So.
Okay, so the next question comes from Peter Lopez. Now, so this is a bit more serious. Could you predict how the utilization of flow cytometry is going to go in the next 5 to 10 years? Going to go up? Going to go down? Or stay the same? Gosh, it's like play your cards right. Higher, lower, stick. Pratip.
It's a hard question, but I think that it's probably going to stay the same because we're going to have more technologies to choose from. And so there's going to be a little bit of a drag on the number of people doing flow. But the flip side of it is that actually doing a flow cytometry experiment is infinitely easier than it used to be. And data analysis is something that we're all focusing on. And so I think that's going to make it easier to take up flow. So those two trends are probably going to cancel each other out.
Okay, and go ahead, Jessica, go.
Sure, yeah. I mean, I would probably challenge Peter to say, like, what do you mean by up, down? I'll give him the engineering answer, which is, that are you talking about how much more use, or in what way is it going to go up? Because, yeah, that's hard to answer.
Okay.
Yeah.
Paul?
I don't think there's any question that the utilization will increase. We do see changes. We see, maybe CD4s are no longer needed as much as they were. There's definitely a transition with MRD going more to next-gen sequencing. But there are always new techniques coming out, new applications coming out. And, we're moving into a world of microvesicles. And I just see that as continuing on for a long time.
So my follow-up question to that. Is conventional flow cytometry dead? And is spectral the only way forward? 5 years' time, is anyone going to buy a non-spectral system? That's a quick question. Yes, no. Jessica?
No, it's not dead. We had a good example of at the meeting with resource-poor places where it's so critical to still use it. So, the complexity, of spectral, whether you want to say it's complex or not. There's still a lot of places in the world that need just, the standard, simple method. I don't think it is. Maybe, 20 years out, 30 years out. Eventually it might get there, but not yet.
Okay. Pratip?
Was it Mark Twain said rumors of my death have been greatly exaggerated? I think that's applicable to flow cytometry because, we thought that with the emergence of mass cytometry, there was no reason to do flow anymore. People are doing flow still. We thought with the emergence of CytSeq and, high parameter protein analysis through CytSeq, we wouldn't have to do flow anymore. And we still do flow. And so, I think it's here to stay.
Okay.
I'm a big fan of spectral. So my first thought was, spectral's going to take it over. But then as you think about it, there's absolutely a lot of applications that are just one color. We're really maybe not all that aware of it. We sort GFP and that type of thing. But out there, you think about the beer industry, and there's a lot of people that are looking at yeasts and using instruments for that. Within the there's a whole lot of sorting going on sperm, for sexing. You hear about places that have 1,000 instruments just running these things. And then, within the milk industry, looking at mastitis. It's so, while I honestly think that where everybody in this room is going is spectral, there is a lot of industry out there that will be single color.
Okay. So what is your favorite tool or instrument in the lab? And I'll start with Paul.
Oh my gosh.
Not a cytometer. What is your favorite tool or instrument other than a cytometer in the lab?
That's a tough question. I know that I had a couple working in the lab, and at night I think their favorite instrument was the centrifuge, and that was for the anyway. I would imagine but the one that comes to mind is the 8-channel pipette, but I have really no real answer. I think it was the idea of just doing the single and being able to move things in 8 channels. So that's a.
If you had an 8-channel pipette, do you think a politician would look really cool? And you think, my God, they can actually hold a pipette. If it's an 8-channel one, they must be really good politicians.
Right, yeah.
Pratip.
Those little microfuges that are like this big that you spin the microfuge tubes in, and you just close the thing and you don't have to press anything. As soon as you close it, it spins like a maniac, and you've got the plastic cover on it, and so you can watch it spin and slow down. If I weren't a scientist, I would just enjoy the heck out of that. So, children's toy maybe.
Jessica.
I have to say the oscilloscope. I just get so giddy when I'm tweaking that and teaching students how to do it. And look at this waveform, and you measure the velocity and full width, half max, and they're like, this is weird. I'm a chemical engineer and I shouldn't be learning this, but yeah. So I the oscilloscopes and the function generators, we can play around with different types of ways to modulate the laser, and it's fun.
So thinking about oscilloscopes, I've got to ask, what was your favorite flow cytometer of all time? Jessica?
Wait, say that again?
What was your favorite flow cytometer of all time?
All time, the Fax Vantage, hands up. Yeah, hands down. I mean, the Fax Vantage for me has been so fun to work with. I can I can open it up, unplug from the PMT, put in new preamplifications. Is that
because it kept breaking? Sorry, Coulter.
Yeah. It's a fun tool to teach students on, and to modify. We do modifications of cytometers.
Okay. Pratik, your favorite flow cytometer of all time?
No comments, because there is no reason for me to anger any of my beautiful ex-girlfriends. It's like, right? It's you say good things about your symphony, and then you're like, yeah, but the Bigfoot is awesome. But yeah, I really had good times on the Coulter in 1996 or something, that I learned flow cytometry on. So, there's no answer to that question. Nice try, though.
I fell for that.
I'd have to go with the Epix 5. That was one of my first instruments, and I love being able to play around, and you could pull out filters easily. You could do a lot of manipulations with that. That would be my favorite.
MoFlow Legacy. Come on.
All right.
Did you not have a MoFlow Legacy?
Was that the really big one that had like a table that went all the way around?
And the telephone switchboard on the side. So when the engineer came, you could just turn things around and completely confuse the hell out of them for ages. John, what was your favorite?
What's the Legacy?
Legacy. Who had hands up if you had a MoFlow Legacy? Now, keep your hands up. Put your hands down if it wasn't your favorite instrument. There you go. See, look, all the hands still up. So, Beckman Coulter, can you please bring back the Legacy? But can you just let it do six-way sorting as well? Okay, so the next question from Claire Brown. What do you most love about your job? And I'm going to start again with Jessica on this one.
To say the flexibility. Just being a faculty, you can create your own hours, design your own research. You interact with students every semester. They're always, it keeps you young. Yeah, flexibility.
Okay, and I'm going to go to Paul next.
So for me, it was working with the students. No question at all. I enjoyed really working with the students, talking about their research, going on, about different experimental design, reviewing the data. There was all the hassles and politics and everything else that was going on, but you could sit down and talk science with the students, and that I love the most.
Okay.
No question about that.
Pratip?
I think being in the lab alone late at night, finishing up a set of samples that, on the cytometer, and you just blast horrible music that nobody else would tolerate. And, and you're in a zone then, and you're just, the experiment's done, and you're just working through, and you're playing with the results on the screen. It's great. It's like, that's my heaven. Well, sort of. Yeah.
Okay, so this is a difficult question set by Evan Jellison. What accomplishment in flow cytometry are you most proud of? I'll start Pratip this time.
This is an easy question for me. It's the introduction of the high parameter, really high parameter flow, doing the going into Mario's office with the first results from a 17, 18 color flow panel and just showing it to him. And I did it just kind of on a lark, and his eyes lit up, and he was like, "You just did the first 17 color experiment." And you can't get those moments in science just as a young scientist where your hero is telling you did something cool, and that was the the start of what I built my career on. So.
And Paul?
Well, I was very early in my career. I was working at a company called Synaxis, and they made the lipophilic tracking dyes, the PKH dyes. And I was working down at the NIH, which in itself was a real experience in Steve Rosenberg's lab. And I was staining up tumor-infiltrating lymphocytes and injecting them back into mice and there was a microenvironment with the tumor, and there was IL-2, so they were obviously stimulated. And I was pulling them back out just to see how many got there, and these damn things were dim. And so I do another experiment, stain them up, even brighter, put them in, and they'd come out and they'd be dim. And I probably did this 10, 15 times. And somebody then turned to me and said, well, Paul, maybe that's a good thing. Maybe they're dividing. And I went, and actually out of that, it wasn't something that I developed, but the companies in Axis then went on to develop something called the Cell Census Plus assay. Now that was looking at dye dilution and proliferation. Subsequently, CFSE came out, and that sort of took a good part of the market. But I think getting into the proliferation was something that I look back on as being really proud of. Yeah.
Thank you. Jessica?
I mean, I might be inclined to say something about my research, because I do fluorescence dynamics and design cytometers to measure lifetimes. But the first thing that really came to mind was just, I feel like I've contributed because I've been part of the first all-female executive committee at ISAC. And I feel like I'm helping to be a role model for the society. So.
And I'm going to ask, how have you found your first 6 months, 9 months as president?
Yeah. Mm-hmm. First few months now, and we'll be continue for a couple more years. Yeah.
And how have you found it?
So that's an answer for you.
Right.
Okay, so the next question now is a very similar question, actually. So the answers may be the same or maybe different. So for Melina, what contribution to the Flow Cytometry Society do you think you've made? To the community?
Okay.
What is your biggest contribution to the community of flow cytometry? I'm going to go with Paul first.
There's, I think within ISAC, I think it's what you're referring to, and I think within ISAC, I felt like I've really made some significant contributions. Maybe one that really sticks out in my mind, and this is something that Pratip mentioned at a council meeting, was CYTO Youth. So that was something that CYTO Youth is still going on. That's something that really impacts on kids and makes kids aware of really what's going on in flow, and really gets them interested in science. And it was something that we developed in Buffalo, where we brought kids in from the high school. We actually put a flow cytometer in the high schools. And the schools. So in 8th grade, they were learning, or 5th grade, they were learning about health, and we would talk about CD4s and demonstrate some of that on the flow. And then in 8th grade, they get it again, and we get into physics. So there's a couple things, but I think Cyto Asia is another one that I was particularly proud of. But Cyto Youth and the way that's continued is good.
Okay, Pratip.
I think it's an unusual answer, but it's the spirit that I hope I'm bringing to working with people in the flow community. That it's, I'm not trying to keep anything for myself, or I'm just trying to be open with what I know and what I understand, and be eager to help. And that was something I learned from Gary Nolan and from Mario, and it's a great way to generate really positive feelings. And so when I walk around here and I get to see people that I've worked with, it's genuine, the happiness of interacting with them. And that's one of the reasons this meeting's so much fun to come to.
Thank you. And
Yeah, and just real quick, I mean, I think I kind of sort of answered that in the last question, but the cytometry community goes far and wide and deep, and I think, I can think of a span of things that I do. I really appreciate working on the associate editorial board for the journal, and then doing things like serving a term in the Cell and Molecular Technology Study section for the National Institutes of Health Center for Scientific Review, because that, you're looking at all of these really novel types of applications and new technologies, and helping, contribute to those becoming something. So.
Okay, so actually, I've just been WhatsApped. Derek, where are you? Question to the panel. What city would you love to have CYTO in? 3, 2, 1, Pratip. Quickly.
Some beach destination. Like let's go crazy. Let's do it in like Fiji or something. Something
Fiji, yeah.
really insane. We'll do it on a beach. We don't need a convention center. Save some money.
Yeah.
Yeah.
Okay.
Oh, I always wanted to do it in Hawaii. I mean, that was
That's what I was going to say.
that was always high on my list.
That
Yeah.
is so predictable, though.
Well, I'm sorry.
So Jessica, what are you going to say?
Oaxaca, Mexico. I know. No, when you had the beach idea, I immediately thought
Hawaii,
Honolulu. Yeah, it would be
Yeah,
fun.
it's certainly been discussed.
So Hawaii. Who would like to go to Hawaii? Why aren't we going to Hawaii? Edinburgh, for goodness sake.
There
Why are we
was
going
a
to
time
Hawaii?
when there was a time, wasn't there, Jessica, when Honolulu was on the list?
It
Yeah,
was-
it's definitely been on the list, and it's
Yeah.
been discussed.
Yeah.
We only knew the president.
Yeah, right. We got the money.
Okay, so actually, I've got one question from Alfonso. Where's Alfonso?
There we
Hello,
go.
Alfonso. So the question is, if you were not doing what you do today, what kind of job do you think you'd be good at? I'm going to go Jessica first.
A mother. I mean, but that's an easy answer. I am a mother now, and I have 3 kids, and But if I wasn't working, just home and hanging with the kids.
Okay.
Oh.
Pratip, you're not allowed to say father.
Sorry?
Yeah, I don't have to say father.
Yeah, no, I'm not going to say father. My kid's going to kill me, right?
And it is being recorded.
It would be, yeah, I know, really. Oh, big mistake. It would be a journalist, and I'm proud to say that my daughter is going to go to school, to college in the fall to be a journalist. So, at least there's some, genetic influence, I think, that's taking over, so I can watch from the sidelines.
Cool.
I'd like to be a naturalist, take people out on hikes into the wilderness and be there identifying plants and flowers and geological formations and stuff like that. That would have done me very well.
You know what? I think I'd like to be a TV host.
You'd be very good at it.
So we are coming to the last few minutes. So I'd like to actually bring up John Tiggs, who's joining us later to be our DJ. John, come on up. Bring the chair forward, please. Bring it forward. So John, the same question to you, because this is no longer obvious, is it? If you could do any other job, what would you do?
I'd be Peter O'Toole.
He died.
Oh, that's right. Peter died. I guess the obvious answer is I would just be a DJ. Yeah. Music's passion, flow cytometry is what I'm good at.
Okay. So I'm now going to go for some quick questions to finish the night. So I'm going to start. This time it's going to go Paul, Jessica, Pratip, John. Who is the best flow cytometrist of your generation?
Of my generation? Of my generation. Well, I think I am. I mean,
I can see what the answers are going to be now. Jessica?
This is hard. I think Pratip's my generation. Pratip.
Well,
Say you.
yeah, Jessica. I mean, I can totally cop out here, But I think that this isn't a fair question, because we are all doing really, really cool work in our own fields, and we are each our own stars.
there you go.
How's
Okay,
that?
all right.
That's good.
And John?
There's no way to follow that up. Because any answer now seems very ridiculous. I agree, right? Pratip, very good. You've made it easy for me. So I agree with Pratip. We all are individuals and great in our own way.
Okay, so this is being recorded today. So I'm now going to go from that side to this side. What is your favorite streaming device?
My favorite streaming
Yeah.
device? What do you mean, like as in an iPhone or a
Yeah,
TV
we're
or
streaming.
service?
Yeah, come on, what's streaming? Come on.
It's probably the computer, because then I can leave it on and watch true crime. Actually, fascinated by it, and I just leave it on in the background. Like those nights I don't want to turn up the music in the cytometry, I find out why that lady got strangled. So yeah.
Jessica?
Probably my iPhone. I mean, I'm really connected to it. I'm surprised I don't have it with me right now.
Okay. Can't believe no one said Netflix. No one said, I don't know, iTunes. Paul,
We've got Oak Creek flowing by the out in the backyard. So I like Oak Creek, watching Oak Creek go. But I actually, to your question, probably I spend more time on the TV, just running streaming on the TV.
okay. And John, what would be the right answer?
The right answer is I would go what we said before, the MoFlo legacy.
That was a setup.
Spoiler alert, Paul. I have the cheat sheet.
Okay, so next question. I'm going to start with Jessica this time. And I'm going to go, what's your favorite color?
My favorite color? I guess. I like blue, but are we going down to wavelengths?
Okay,
yet? What are we
blue,
doing?
blue's good. Blue, oh, blue. Oh, crap. We're all wearing blue. Blue,
Yeah.
405.
405.
PE.
And,
Fluorescein.
yeah,
Okay.
okay. Okay, and on that note, I think I have covered most of the questions for tonight, and we are bang on 40 minutes. So I'd just like to thank everyone for actually being really quiet. It's been really fantastic to have you here. You've been engaged. Thank you for sending the questions. A big thank you to our guests tonight.